Okay, good morning, everyone. As the numbers continue to rise on the people attending, I think we'll make a start just so that we're in good time for the end of the webinar. So welcome to the introduction to Codecs, code detection by indexing webinar. My name is Paul McMillan. I'm the manager of the Center for Advanced Histology and Microscopy at the Peter MacCallum Cancer Centre in Melbourne, Australia. Before we start the webinar today, I'd just like to take a short time just to acknowledge the traditional owners of the land on which we're meeting, the Kulin Nation, and pay my respects to their elders, past, present, and emerging. So this started out as an internal webinar to highlight the fact that we have the Codex system on an extended demo until the end of the year here at Peter Mac. But has since expanded out quite a bit, and now we have attendees from across not just Australia, but also Asia Pacific as well. So thanks to all of you for coming along this morning. Let's see if we get my slides to move. Okay, so while I have you, just a very quick word about the center. So we have 4 platforms within CAM, as we call ourselves. So a histology platform, an optical microscopy platform, an electron microscopy platform, and also an image analysis platform. And the team is made up of 8 team members. So we have Danya, Basia, and Leah, who take care of our histology platform. We have Judy and Alicia, who take care of the electron microscopy platform. Rayhan deals with our immunohistochemistry platform, so is involved in the Codex system. And we also have Meta, who's our optical microscopist, who was also involved in the Codex system. If you guys want to get any more information at any point, then just use that cam@petermac.org email at the bottom there. Okay, so let's move on to today's program. So this is the brief introduction from me. We're then going to move on to 3 presentations, one from Dr. Grady Carlson from Akoya, giving us a brief introduction into Codecs. The next one is from Dr. Oliver Braubach, again from Akoya, giving us an overview of single-cell Codecs applications. And then finally, we have a more research-based presentation from Dr. James Wilmot. Sorry, James, I knocked off your doctor there at the start. And he's going to give us an overview of what he's been doing so far with their Codex system at the University of Sydney. At the end, we'll have approximately 10 minutes for questions and answers. And we've got a couple of people online from Akoya Australia and also Akoya Asia Pacific to answer those questions. Okay, so hopefully you guys can see Grady's presentation. So I'll just give him a quick introduction. So he's the bioinformatics field applications scientist supporting Codex image analysis and was previously with PerkinElmer and also had roles as a postdoc at Baylor College of Medicine.
Hello everyone, and thank you for having me here today. My name is Grady Carlson and I'll be providing an overview of the Codex technology. The Codex technology was created to address the need for multi-marker spatial analysis. Now, the need for multi-marker spatial analysis became apparent as a number of publications began to show that it's not only the composition of tissues that affects biological outcomes, but it's the spatial proximity of the cells within those tissues that affect biological outcomes. And this tells us that we need to know not only what cells comprise that tissue, but the location and the relationships between the cells in that tissue. So here's how Codex enables us to get multi-marker detection of cells inside tissue. We start out with a cover slip on which a tissue is sectioned. We then incubate this cover slip with a cocktail of DNA barcoded primary antibodies. This cover slip with all of the tissue and barcodes bound now goes into the Codex insert, which sits on the microscope stage. The integrated instrument is fully automated, so there's no manual application of Codex reporters or Codex reagents. This automation facilitates the imaging of all of our markers inside of the tissue. So let's take a deeper dive into exactly how this workflow allows us to acquire all of our images and target markers inside of our tissue section. The tissue is stained with all of the antibodies at once. And this is because we have a cocktail of DNA barcoded primary antibodies, so there's no issue of cross-reactivity. This cocktail is stained on the tissue and the antibodies are fixed, so they will not be removed. At this point, there are no fluorophores on the tissue. Just these DNA barcodes on the antibodies. Now, as I mentioned, these DNA barcoded antibodies allow us to bind fluorophores with the complementary oligonucleotides. Now, these reporters, or fluorophore-bound oligonucleotides, are going to enable us to detect where our antibodies have bound to the tissue section. So as I mentioned earlier, the workflow for Codecs is to reveal the antibodies that have bound to the tissue by introducing these Codecs reporters. We're then going to use Codecs automation to take a picture of where these reporters have bound to their target antibodies, and then we'll remove these reporters so that we can repeat the process of reveal, image, and remove. This process allows us to acquire all of our markers with Codecs. So Codecs has a number of components. We have the reagents, we have the automation, which includes fluidics and microscope automation, and we have analysis. So Codecs reagents include all of the kits and reagents needed for running the instrument and the fluidics. The Codecs antibodies include a number of different antibody configurations, which we'll talk about later, but they do include inventoried antibodies that can be purchased from Akoya. Codecs reporters allow us to detect those antibodies once they've bound to the tissue section, and Codecs barcodes can be conjugated to antibodies that we do not have in inventory. So I mentioned that I would elaborate on the different types of Codecs antibodies that we have. The validated antibodies are sold as a complete assay. The Codecs screened antibodies are tested for barcode and reporter compatibility, but are not sold by Akoya, and user conjugation is required. And the Codecs community antibodies are tested and shared by Codecs users, but not tested or sold by Akoya, and user conjugation is required. So for inventoried antibodies, conjugation is already performed by Akoya, and for all other antibodies, the conjugation would need to be performed by the end user. So what tissues are compatible with Codecs? Well, human fresh frozen, mouse fresh frozen, and human Ffpe are all compatible with Codecs, and here's some antibodies that have been used in Codecs experiments. Now that's not to say that Codecs is necessarily limited to these tissues. This is simply showing what antibodies we've seen being used for Codecs experiments, and primarily that's on human fresh frozen, mouse fresh frozen, and human Ffpe tissue. So moving on to the fluidics control and the Codecs automation, the Codecs integrates with a microscope to allow for imaging of our tissue section. You can see the fluidic lines feeding to the microscope stage right here. So this custom insert holds the tissue and integrates with the microscope stage. And this insert is now shown where you can see two gaskets are sandwiching the cover slip that is going to have our tissue on it. So that's how the Codecs integrates with the microscope. And tissues are sliced onto the cover slip just like they're sliced on the slides, like traditional histopathology. And staining occurs offline. So staining does not occur on the instrument. We do reveal where the antibodies are with the instrumentation, which is how the reporters are delivered via the fluidics here, and then we image with the microscope and then remove those reporters, but the staining of the antibodies actually happens offline. So just to reiterate the workflow, we're first going to section a tissue onto a cover slip, incubate with a cocktail of primary DNA barcoded antibodies. We're then going to sandwich that cover slip and tissue between two gaskets with the Codecs insert and put that on a microscope stage. this integration is fully automated. So once our staining has occurred, the rest of the workflow is fully automated and involves the reveal, image, and remove process where we can image 3 markers in every cycle to acquire all of our markers with Codecs. So here's an example of a Codex experiment in which 41 markers were imaged on Codex tissue. Again, we're compatible with fresh frozen and Ffpe tissue sections. So in terms of software, we have the ability to use either Codex Multiplex Analysis Viewer, which is an ImageJ plugin, or we could use Visiopharm. Now, Codex Multiplex Analysis Viewer, or MAV, is a multiplex viewer that uses clustering and gating to define phenotypes and includes a spatial analysis tool to calculate spatial metrics and investigate spatial relationships. And Visiopharm is a streamlined solution for high-quality cell segmentation, high-dimensional phenotyping, i.e. clustering, batch analysis, and multiple spatial analysis tools, including cell neighborhood analysis. I'll talk a little bit about Codex Multiplex Analysis Viewer, and I'm happy to talk about Visiopharm with anyone that's interested in talking about using Visiopharm. Okay. So with Codex data analysis and MAV, all of the data come into MAV post-segmentation. So the segmentation actually occurs in the Codex processor and is read into MAV. And here we can see that the segmentation file includes X and Y spatial coordinates, so we know where each cell is inside the tissue, and it also has columns for each marker or antibody, so we have all the cytometric information about each cell and their spatial location. There are multiple methods of analysis available inside of MAV. Thresholding and gating is one, where we can, just like flow cytometry, select the cells we want by gating on the regions of interest, or gating on the populations of interest, rather. There's clustering and t-SNE, which allows us to cluster either inside of MAV or externally and then import the data back in. We can use ROI to select regions of interest, and spatial analysis to calculate spatial proximity and the likelihood of interaction. So this is an example of Codex MAV where we're thresholding. You can see a couple of different things inside this slide. So first we see here that we have a tissue that's been stained with CD34 in red, and then I've detected where there's CD34 positive cells, and that detection is apparent because of the green dots, which are overlaying the cell nuclei. And so how were these green dots formed? Well, first we came into the DAPI versus frequency plot and recognized cells that have positive DAPI. Then we came into the CD34 child plot and selected the cells that had high expression of CD34. Here the red and the green are different colors than the blue because they have high expression of CD34. So we're seeing the dynamic range color scaling of the CD34 expression in this plot, and I've selected the upper range of expression. So this is a typical view inside of Codex MAV and a typical gating workflow. MAV also has the ability to cluster cell populations, and the benefits of clustering are that it has relatively high throughput and it enables complex phenotyping and discovery of novel phenotypes. Of course, all this high multiplexing with Codecs then enables advanced phenotyping and spatial mapping, so we can then understand where these complex phenotypes are inside the tissue. Here there's a Verona diagram showing both the color and letter annotations of the different cell types inside the tissue. For this follicular structure, there were over 20 cell types. And lastly, we're able to use the Codex Multiplex Analysis Viewer for spatial interactions. So understanding the likelihood of spatial proximity, the number of interactions that we're seeing. So red indicates a high number of interactions between follicular dendritic cells and B cells, whereas blue indicates a weak number of interactions or a low number of interactions between vascular endothelial cells and B cells. And so it's these spatial interactions that allow us to draw those biological conclusions that we wouldn't be able to see without Codecs. Well, thank you for your time. And if you have any questions, please contact us.
Okay, so I'd say thank you to Grady for that presentation. Feel free to stick any questions that you might have into the Q&A or into the chat. What we might do is hold those and answer them at the end as a group. But yeah, as you think of them as they come through, please don't hesitate to stick those questions through. We'll next move on to Oliver's presentation. Just let me see if I can get this sorted out. Share screen. So before I start the video, we should just introduce Oliver Brobach is the manager of the Akoya Applications team. He has a PhD from Dalhousie University and had postdoc appointments at Yale and the Korean Institute of Science and Technology before joining Akoya.
Hi, everyone. My name is Oliver. I lead the applications and innovation at Akoya Biosciences. And I'm going to follow up Grady today by talking to you about single-cell spatial phenotyping. This is our concept that we have at Akoya, and that fits in the many applications that you can use this system for. What you see on this slide is advanced breast cancer, a stage 2 adenocarcinoma, been imaged with a Codex system. Tissue is about 2 millimeters from bottom to top, 3.6 millimeters from left to right. And what you can see are 4 different labels. Now, I'd like to start my discussion just by showing you this image and reminding you that Codex is optical imaging based and uses epifluorescence imaging to detect signals. Hence, we have single cell resolution. Very nice. You can see a nucleus right here. If you can see my cursor, you can see a nuclear envelope in red, which is laminin B1. And you can see E cadherin, which is presumably the plasma membrane of this cell. So not only with Codex do you have the ability to image large, intact whole tissues, but you can do so at single cell resolution. And then as Grady has alluded to, of course, you can do cyclic imaging of many biomarkers on a single tissue. So as you can see, the slides are advancing. We're always looking at the same tissue, but we're always looking at different marker combinations. These are toggling down here on the bottom. Each time you're looking at 5 to 7 different markers on the same tissue, but each time you're seeing a very, very different biology of this tissue. That is Codex single cell spatial phenotyping. You have single cell resolution. In some cases, you can go down to subcellular resolution, and you can look at your tissue with up to 40 or more biomarkers. Now, how does this concept of single cell spatial phenotyping work? Essentially, what we do is we register every cell by multi-parameter segmentation. So you can see, as I'm going back and forth, here's your actual image, your actual fluorescence image, and here's a segmentation map. Every cell has now been registered as an object. What happens once you register every cell as an object, of course, you can start to catalog it. Every cell has a number. In this particular tissue, there are 63,000 cells. Each cell will be labeled with a combination of antibodies. In this particular experiment, once again, we labeled with 36 antibodies. Every antibody will label every cell with a unique combinatorial pattern. So each cell will be labeled with these antibody combinations and unique combinatorial patterns. Cell 1, for example, will be labeled, somewhat with antibody 1, but not with antibody 2. Cell 2 will be strongly labeled with antibody 1 and somewhat with antibody 2, and so on and so forth. These data are analogous to data that you get, say, in single-cell sequencing or even a flow cytometer. The difference in Codex data, of course, are you have XY coordinates. You know where every one of your cells is in X and Y. So this you can see here for 36 biomarkers, 63,000 cells. Now, what we do is we use clustering of data. In this experiment, we used unsupervised algorithm to cluster your data into different phenotypes. With the data that I had just shown you, we were able to identify 12 different phenotypes. You can see these here on this t-SNE map. Every dot is a single cell, And every color represents a different cell phenotype. That was identified via clustering. You can see a very large contingent here of sort of green cells. Those will be luminal cells, a type of epithelium. Down here in blue, you have another type of epithelium. Up here will be vascular cells. I know these things because I've looked at these data many times over. But what I would like to point out to you is this small contingent here of red and orange cells. Out of the 63,000 cells in this experiment that we had looked at, 546 of them are so-called basal cells. These were identified with Codecs. Now, 546 of 63,000 is 0.8% of the total population. That's a pretty small amount. Even better, there are 44 luminal cells in this particular tissue. That's 44 out of 63,000 cells. That's 0.07% of the entire cell population. Now you can see an example of these luminal cells right here. So this is an epithelium, so secretor epithelium, and nudged in here are these three little cells. There are a couple more over here, and here and there you see additional ones. How can we identify these? Well, evidently via clustering, the algorithm picks them out. You can look at a heat map and you can see that these luminal cells, for example, they express Ffpe, whereas basal cell, which will be probably the next most similar cell, if you want it that way, does not express Ffpe. On the contrary, the basal cell expresses TP63, whereas the luminal keratin 814 cell does not. Beta-catenin is labeling these cells, but not this one. And you can go through your heat map and you can look at it. And this is, again, very similar to something that you would do in, say, single-cell RNA sequencing. We are basically looking at the outputs of clustering runs, and you're trying to make sense of it, trying to see whether or not a cell is there. The nice thing about this experiment, the Codex experiment, though, is you have visual confirmation. You can actually see your cells. Here's 3 of them there, a couple dozen up here. There's a couple more here and here. You can actually see the cell. You have the clustering data, you have the heat map, you have the cell right in front of you. You can touch it almost. There you go. This is single cell spatial phenotyping. It's one way of looking at your tissue down to the single cell level with a high biomarker plex and even being able to identify the very rarest cells. There's a single one of these cells here, 3 of them over here, a couple I pointed out here. This is incredibly granular, incredibly detailed way of looking at your tissues. This is an application that can be done in any type of tissue for any type of biology that you're interested in. It's very reliable, clean detection of different cell types. So one of the other things then to touch on is, what do Codex data look like? What does single cell Codex data look like? I'm just going to give you a primer on this. What you can see here is another different Codex experiment that we've done. In this case, we labeled a human tonsil with 39 different biomarkers, antibodies. Each one of these images shows you a representation of this very same tonsil. Each time, it's labeled with different antibody combinations. You can see here, for example, this panel, a lot of B cells labeled with CD20. You have the T cells outside of these sort of circular follicles or proliferating regions for the B cells, and IDO1 labels yet another structure in this tonsil. So the data add up nicely. They're nice to look at, but they just have a wealth of information here. Now, assuming your experiment works out nicely, what you do next, or what we like to do next, is we like to have good tools and able to make sense of them. For that, we go back to tools that are being used by single-cell biologists all over the place. In this case, we look at protein co-expression. For example, here, you can see all of the 39 biomarkers in the experiment that we've conducted juxtaposed with 39 biomarkers from the same 39 biomarkers. And when you have this diagonal red line, basically that means you have perfect co-expression of a biomarker. Hence, a cell that expresses CD45 rho should also express CD45 rho, and therefore it's red. But if you look down, if you walk down this diagonal line, you get to see these boxes, basically. And each one of these boxes kind of tells you that a cell that, for example, is expressing beta-catenin is also likely to express E-cadherin, which is in this line, or pancytokeratin. And within this box, you realize that all of these are different epithelial markers. So you get a squeaky clean cross-correlation of epithelial markers. Vascular markers, same thing. Nice little box, CD31, CD34, collagen, SMA, and so on and so forth. These are real Codex data, about 350,000 cells in a tonsil. Cross-correlation works really nicely. The T cells up here, the vasculature here, you have B cells here, epithelial cells. This is one way that you can analyze your Codex data. Now, of course, and I've just already shown you some clustering runs, once you have your data, once you have it analyzed, once you have the QC done, and you think that your expression patterns are looking good, you can cluster your data. And what we have done here, we clustered data from a tonsil. Identified approximately a dozen different cell types. You can see them here. I'll tell you how many of them there are, 2% fibroblasts, some macrophages, myeloid cells. And then over here, you can see how these different biomarkers are expressing. So this, for those of you who don't know, this is a UMAP. So this is a reduced dimensionality representation of all the cells, once again, in your tissue. Each dot is a cell. Each color indicates the cell type. Yellow, for example, your natural killer cells. Green, your B cells, and so on and so forth. You can compare that to the actual expression patterns of the markers on your UMAP, where red is hot and blue is cold. So for example, IDO1 expresses very strongly here. You can go back to your map and you'll see that this is type of epithelial or T cell, depending on what color this is, and so on and so forth. So this is not too much different from what you'd be used to if, say, you're doing RNA-seq. But where it gets really different and really exciting with Codecs is the idea that you can actually take these cell phenotypes now, these are the cell phenotypes we've seen, and you can map them back on a tissue. So what you see now here is a piece of breast tissue again. So secretor epithelia here, these sort of rose-shaped regions, and these ducts or lines here are the collecting ducts where the fluids get collected. Now, I'm not showing you a microscopic image. What I'm showing here is a phenotypic map, where each dot, each color is a cell of a certain cell type. T cells, dendritic cells, endothelial cells, fibroblasts, and MACs. Now, if you look at that a little bit more closely, you can see it's quite a jumble of different cells. Generally, you have a lot of luminal cells, secretor epithelia, surrounded by basal cells, but there's also a contingent of immune cells inside of this breast. And here's where really this type of single cell biology differs from all other types of single cell biology. That is that we know the connectivity of every cell with every other one. It is very easy to draw this map. It's very easy to know that this cell is, many dozens of connections away from this cell, and then so on and so forth. But having these data now allows you to look at your tissue and to ask questions that you've never really asked before. For example, we find Tregs or CD4 T cells in the breast tissue. This will be this little red cell right here. What we've done is we've gone around and we asked throughout the breast tissue, if you find a single T cell, CD4 T cell, what is the likelihood that it will identify another T cell? And there's a certain neighborhood cluster. There's a certain part of the breast tissue that the likelihood's quite big. And that will be cluster number 1. This is what you can see here. Each time here, you can see a T cell, CD4, that is surrounded by a bunch of different C cells. This is an actual neighborhood that exists in this breast tissue. It's a place where a CD4 T cell is quite likely to encounter another CD4 T cell. On the contrary, there are other CD4 T cells that are falling outside of this area, and they are more likely to encounter CD8 T cells. Now, these patterns may relate to the density of a certain cell type, makeup of tissue, or all kinds of things. However, what is probably clear and that we are really starting to follow up on nowadays is that no matter what the biological basis of these organizations is, the functional implications of these are very important. Because a T cell that is located to a like T cell will behave different than a T cell, for example, that is located to an epithelial cell that's expressing PD-L1. These are very, very different scenarios, and they very much decide on what this T cell ends up doing at the end of the day. So analyzing these types of neighborhoods is something you can very easily do with the Codex experiment. And if you have never heard about this before and you're interested in it, I encourage you to read this paper by Christian Schurch and Gary Nolan. This came out late last year. I think this was the first time that this concept has been introduced, and they do a really nice job here in showing how these types of neighborhoods can really relate to different patient outcomes in, say, colon cancer. A really cool paper, and I think this is sort of really the study that has grandfathered this approach of these cellular neighborhoods. So that's just one example that of an application that you can do with Codex imaging. So you can do cell phenotyping just like you're used to from all the other applications that we see nowadays. But now instead of just knowing which cells are there and how many cells are there, you can ask all kinds of new questions like who are they neighboring with, how may they interact with, and ultimately, how could this affect or relate to a patient outcome or a certain type of disease. So I don't have a lot of time, and I'm going to just wind up this little segment of mine with sort of a section on open Codex applications. So I gave you sort of a conceptual overview of what you can do, but I'll give you a much more practical overview of 3 examples that certainly you can do as well. Obviously, Codex is for whole tissues. Now, I'd like to emphasize this once again. When you take a Codex experiment, you're imaging continuous whole tissue. You can do 12 by 12 millimeters on our current cover slip. That's a big piece of tissue. So there are no ROIs. You're not restricted to predetermined ROIs or post hoc analysis of certain ROIs. You have the whole tissue at your disposal at single cell resolution. That's definitely one of the strengths of this approach. You can also do tissue microarrays. Well, you can see here, these are biopsies from lung tissue or imaged in a Codex experiment. Now, this isn't the entire experiment. In total, what we did in this experiment was something like 54 cores. I'm showing, I think, a fraction of these, but it's just as easy to image tissue microarrays as it is to image a whole tissue. The only limitation, the only rule here is you need to be able to fit them on your cover slip. Lastly, you can even image cell cultures and organoids. What you see here is a neuronal cell culture. These cells were grown directly on the cover slip. They were fixed, and then they were labeled with antibodies and processed for our imaging experiment. The cells did not wash off. We were able to look at this culture with, I think, 36 different antibodies, seeing things that we never really seen before. See here glia, see neurons, see all kinds of connectivity between different parts of the culture that we had never really understood. We don't even understand them yet, but we've never even seen them before. So the application range of Codecs is essentially anything you can image, you can do your Codex experiment with. Now I just ask you to imagine, okay, I've done IHC with 3 markers for much of my career. What am I going to do with 42 markers? What am I going to ask? What question am I going to ask? How am I going to phenotype my cells? How many cells are there? Where are they? How are they interacting with each other? These are questions that we ask with Codecs. And once again, any tissue, any organ that can be imaged, in principle, can be used for Codecs. We've done this on melanoma. We've worked with brain tissues, human Ffpe postmortem, I should say, which are very difficult to image, but they work just fine on Codecs. Lung, skin, gut, kidney, even dissociated blood cells, spleen and breast, you name it. We've tried it, and so far, this has been an incredibly versatile system. And I hope you find a use for it. I hope you found this exciting, and we're happy to address any questions that you have. Please contact your local reps. Thank you very much.
Okay, thank you, Oliver, for that presentation. We will move on and again, continue to put the questions through. There's lots of good questions coming through. It's probably easier if you put them through the Q&A rather than the chat. It's easier for us to keep track of what's been answered and what hasn't been answered if you go through the Q&A. Our next speaker will be Dr. James Wilmot. Dr. James Wilmot is an NHMRC Investigator Fellow based at the Melanoma Institute Australia in Sydney. He has his BSc and his PhD from the University of Sydney, and he's going to give us an overview of his work on the Codex system so far. Over to you, James.
Right, today I'll be talking to you about how we've been using the Codex multiplex imaging system and how we've been trying to combine that with single-cell RNA-seq data to better understand the tumour microenvironment of patients treated with immunotherapies. It's a dual presentation, so I'm doing a lot of the image analysis and I love the pretty pictures, whereas Dr. Camelia Quirk's a computational scientist in the group who takes the FCS files and the big data and makes the amazing plots with them. So it's a combined effort. So just an overview, I'd like to raise some of the clinical questions that we think we can answer with these type of technologies. And the two technologies we're trying to combine are the Codex system that you've been seeing today. We've been trying to combine that data with single-cell RNA-seq data on the same sample to bring the two technologies together. We think it's going to be a really powerful tool to understand the tumour microenvironment. And we're aiming to use these tools to understand why patients might or might not be responding to immunotherapies. It's a really important question because in Australia, more patients have died of melanoma last year than have died in the whole entire period of COVID-19. So it's a real killer in Australians. And the majority of patients who develop metastatic advanced disease often die. So it's a major killer in Australian patients. Thankfully, we have immunotherapies, which are highly effective in treating advanced disease. These can be either single agent anti-PD-1 inhibitors or combination of anti-PD-1 inhibitors and CTLA-4. And melanoma has really driven the use of these type of technologies and these drugs. And what we've found is while it's highly effective and the majority of patients are achieving a long, good survival, so we can see here out at 5 years, the majority of the patients, 52% of patients treated with the combination inhibitors are alive. So that's a great achievement, but it also raises the important question that there's this up to 48% of patients are dying within 5 years, even with these highly effective treatments. And that's compounded by the fact that the majority of patients treated with these inhibitors will still recur, the disease will come back, and these patients need another type of therapy to try induce a long and durable response. So the two burning clinical questions we have in treating melanoma patients with advanced diseases, when a patient comes in to see the oncologist, is that patient likely to be one of those lucky 50%, or are they going to be in the 50% that need some other type of therapy to save their lives? And that's a major question we've been tackling. We've been using different things such as predictive tests or models, using clinical data, genomics, gene expression, and pathological information, spatial pathology, so tumor immune profiling. We're combining that into predictive models to predict who's likely to respond and not respond to therapy. That's not really a part of this talk, but it's something we're focusing on a lot. But the other major question is, if you can identify which patients are unlikely to respond to standard therapies, how do we know which drug those patients should get? So if they're unlikely to respond to standard of care therapies, which of the many different inhibitors should that patient get to achieve a durable and long response for their particular tumor? And what we want to be able to do is move away from a flip of the coin of the patient achieving a durable response to somewhere where the majority of the patients, or nearly all the patients, are being cured. It's a major problem. Even if we can identify which patients are unlikely to respond to the standard of care therapies and need another type of therapy to save their lives, there are many other, there's multiple other agents that are able to be given to these patients. So at the Melanoma Institute Australia, we have over 27 immunological agents in clinical trials, in over 50 clinical trials. So picking which drug is right for each patient is a major problem facing clinicians, and it's something we're trying to address. This is really important because we've seen studies, large-scale phase 3 trials, where patients were given combination inhibitors in an unselected manner, so all patients given the drug regardless of their biomarker expression. And this has ended up to be a negative trial. However, we know that things such as IDO are uncommonly expressed in some patients. So in metastatic melanoma, as little as 6% of patients express IDO in their tumour. So it raises the question, what happens if we conducted these type of trials where IDO inhibitors are only given to patients that are IDO positive? Could we achieve a much better survival for a subgroup of patients? And to do that, we need to be able to profile multiple drug targets within a single piece of tissue, and as well as understand which cell these drug targets are expressed on to try and understand how they might be, how the drugs might affect the tumour microenvironment. And profiling a tumour microenvironment is a really important question because we're lucky that we have multiple agents that we're able to give to patients. However, knowing which agent is right for which individual patient is a major problem. We have multiple T cell and NK cell stimulants. We can hit the antigen presentation machinery of the tumours to try and make them more visible to the tumour. We can hit the stroma and vasculature to help remove the support for the tumor, or we can hit the tumor itself to try and induce drug-induced tumor death. So with all of these different mechanisms, we need to know which one is right for each individual patient. And we need some technology that we're able to profile all of these in a single piece of tissue and understand which cell they're on. So we turn to the Codex system as one of the technologies we're using to profile the tumor microenvironment. This is Zara Yazin is the research assistant in the lab running the machine, and our system's a Codex box. As you can see on the top image, we have a Zeiss Axia observer, and we have a photometrics BSI camera. And with this system, we're able to take thin sections, so 3 to 5 micron sections of formalin-fixed paraffin-embedded tissue, apply 40 to 50. So I think we've tried 46 antibodies that have DNA barcodes, as we've seen before. We then apply, we incubate the section, and then put the section onto the cover slip onto the machine. Then the machine or the Codex box will detect 3 reporters at a time, wash it off, and repeat till it's imaged all the different antibodies in 3 or 4 combinations. It then exports all the images for downstream analysis. And on the bottom left, you can see just a high-power view of the cover slip well. So the tissue sits in this little well, and the reagents are applied and washed off the tissue to achieve the staining. So one of the first staining attempts we used was a 46 marker panel, which included T cell markers, myeloid cell markers, B cells, tumor cell markers, so melanoma specific, as well as actionable drug targets, so immune checkpoint inhibitors, vasculature markers, and multiple phenotypic markers, so a real broad tumor immune profiling panel. And one of the first questions we had when we were looking at adopting this technology is, are we able to achieve, let's start a movie here. Are we able to achieve high resolution imaging? Because we know if we want to do image analysis, you need very high resolution images to get accurate single cell phenotyping. And with this system, this is one of our melanoma sections stained with the panel. We can see the resolution is excellent, and we can see compared to some of our other singleplex or up to 6-plex staining runs, we can see every immune cell or tumor cell or vasculature within the tumor microenvironment. So really powerful technology that we're able to profile and characterize all of the cells in the tumor microenvironment. So it gave us confidence that we'd be able to take these images and create useful data from them. We then needed to convert the images into data that we could use to analyze. And we used a few different techniques at the time. However, we ended up using the Halo AI system to do our tissue segmentation. And we used the system and trained it to look for tumor regions here in the white, blood vessels in the red, and we're able to train it on the stroma or the normal tissue here in the blue. And with the 40 markers, it's quite easy to train the tissue segmenter. We found the tissue segmentation the cell segmentation was highly accurate using the watershed method, and we're able to get each individual cell accurately segmented. This is really important. If this isn't accurate, then all of the downstream nearest neighbor analysis and all of any analysis downstream is not accurate. We're then able to export out the FCS file into and then import that into Surat or other single cell type technology. So this is where CAM takes over, and CAM has been experimenting with multiple different tools to try analyze the data and understand spatial relationships. So our main goal was to convert the data into unsupervised clustering information where we can identify individual cells based on their protein expression profiles, and then go back into the tissue, which is what is additional to single cell technologies such as single cell RNA-seq, and identify where these clusters are in the tumor. So where are these cells located? So that's a really powerful tool that we're able to do with the Codex system and the data it produces. So our first use of this technology was to try pilot in a really interesting patient that the oncologist had identified in the lab. This patient came in to see our oncology clinic, and they had metastatic melanoma. We had a pre-treatment biopsy before they went on to any of the subsequent therapies. Then the patient was given single-agent anti-PD-1 nivolumab. Unfortunately, the patient's disease progressed and grew on this treatment, and the patient was given a second combination inhibitor of anti-CTLA4 and anti-PD-1. Unfortunately, the patient's tumor grew again and progressed and was resistant to these therapies. We then had a tumor biopsy taken from the patient when they'd progressed on these two standard of care therapies. The patient was then given anti-PD-1 and LAG-3, another novel inhibitor, to try and induce a response. Unfortunately, again, the patient's disease was progressive and grew and resistant to treatment. The patient was then given a lenvatinib, a VEGF and multi-kinase inhibitor in combination with anti-PD-1. And fortunately, the patient had a partial response and a durable response from this last treatment. And then we have a biopsy taken from this patient when they progressed on all of these therapies. However, we have a biopsy taken just after and when they're responding to lenvatinib and PD-1. So this really interesting patient gave us the opportunity to study the evolution of the tumor microenvironment during treatment, try to understand resistance and how that resistance is reversed. We have a portion of the tissue that's put into formalin fixed tissue that we can run through, we can section and run through the Codex system. And the matching lesion, we've also performed single cell melanoma dissociates, where we've got all of the single cells dissociated out into a solution, and we can perform single cell RNA-seq on the melanoma dissociates. So our pipeline was to combine these two technologies, the Codex multiplex system with a single cell RNA-seq system, to try and understand, to try and identify unique and interesting populations in the single cell data, and then use the Codex data to try identify where these cells are within the tumor microenvironment and what cells are around them. So as a path group, we're really interested in getting high quality images, and we found the majority of the markers work very well, and we get images that we're used to seeing with other technologies such as Opal or any of the other immunofluorescent technologies. So we're quite confident with the staining we're getting, and we're able to perform accurate tissue segmentation, cell segmentation, and then export the FCS files for subsequent analysis. Cam was then able to cluster the cells. So we have, in 7 biopsies, we accumulated over 2 million cells. So this is a huge number of cells to process and analyze, and Cam was able to perform unsupervised clustering of the different biopsies from this interesting patient. She's then able to map and anchor these individual clusters in the Codex data back to clusters developed based on the single cell RNA-seq data. And she uses the nearest neighbor weighted analysis to try match markers that are common between the two platforms and assign like clusters to each other. So T cells in a single cell to T cells in the Codex data. And I'll just show some examples of the success of that. So this image showing that on the top, the columns are the individual single cell RNA-seq based clusters. And on the rows, we have the Codex staining for the individual clusters. And as we can see, like we get from our single cell RNA-seq site-seq data, we're able to identify clusters within these images and then investigate the protein expression of the 40 markers within each cluster. And the benefit that we're able to do with the Codex data on top of the single cell RNA-seq data is we're able to go back into the tissue, identify where these clusters are and where these cells are located. So that's really important when studying the tumor microenvironment. So here are some of the images. So we have a UMAP-based cluster here, red showing the different positive cells for the cluster. And below we have the Codex-based image of each of those representative areas. We can see here for cluster 3, based on the single cell RNA-seq, that is associated with melanoma cells co-located in the image below. And highlighting the utility of the Codex system, we can see cluster 5, which represents B cells, which is a large proportion of the cells in this tumor microenvironment. And if we're looking at single cell data alone, we think it made up a major proportion of the tumor. However, when we look at the image, we notice that all of these B cells are in the stromal region, peritumoral region around the tumor and not infiltrated into the tumor. So we can gain spatial context to these cells by combining the single cell data with the Codex data. So going back to our patient, we have the biopsies run through the Codex, and we can see on the top row are the pre-treatment biopsies. The middle row are the prog biopsies. So these are when the patient's progressed on all of the standard of care therapies. On the bottom, we have the patient's biopsy taken when they're responding to PD-1 plus lenvatinib, the vasculature inhibitor. One of the first things we noticed with the blood vessel markers, CD31, 34, we can see changes in the vasculature density and size following the different treatments. That's something we're not able to do with our single cell technologies. So we can see large vessels in the pre-treatment biopsy. However, at the prog biopsy, we can see these microvesicles have developed with very small vessels spread throughout the tumour. However, once the patient's treated with PD-1 plus lenvatinib, we see again large vessels forming within the tumour microenvironment. Within the same section, we can then go look at T-cell infiltrates. We could see in the pre-treatment biopsy, the tumour was, the immune cells were excluded from the tumour microenvironment. When the patient had progressed on all of the standard therapies, there are very little, if any, T cells within the tumour microenvironment. However, when the PD-1 plus lenvatinib inhibitor has been added, we can see a large increase or infiltrate of T cells into the tumour microenvironment following this therapy. So we can really look at the changes in the immune profiles during treatment using this technology. From the single cell RNA-seq data, we identified changes in MHC class 1 expression during treatment. We can see in the top pre-treatment biopsy, large vessels with the tumour endogenously expressing MHC class 1 throughout the tumour with high expression in all cells. However, at the progression biopsy, we can see these microvesicles with very little MHC class 1 expression in the tumour, so loss of MHC class 1 expression, which is the known mechanism of resistance to immunotherapy, with only a little bit of MHC class 1 expression around the blood vessels. However, when the patient is treated with PD-1 plus lenvatinib, we can see a dramatic increase in the size of the blood vessels and a return of MHC class 1 expression, and that coincides with a dense increase in CD3 infiltrate migrating from the vessels into the tumour and attacking the tumour. So we can really understand the dynamics of the tumour biology using the technology. At this stage, it's a work in progress. Cam's been developing multiple different methods to analyse the data downstream, but we're happy with the images we're getting and the type of data. It's just a matter of developing the pipeline to analyse them. Some things to consider when working with the codecs that we've learned with our experience so far is the data size is big. So it can get up to 1 terabytes per slide. So it's something that needs to be considered if you're going to undertake this type of analysis. And when we've been using the Halo AI system to tissue segment and cell segment one image, has been taking about 1 day. So with 18 CPU, 11 gigabyte GPU, it's taking a day to process a single slide. So it is large data. The other major thing when you're going into pathology, when you're used to working with whole sections, is that you can only stain a cover slip in this system. So 22mm cover slip. Of that, you can analyze around 12 to 15 millimeters of working area within the well. So the size of the area you can analyze is not a whole slide. We also encountered some problems with sensitivity of some of the markers. So we originally stained with 46 markers, however, only 40 appeared to work. And some of the markers, such as PD-1, PD-L1, required amplification with the tyramide system. So that might be something to consider with some of the lower expressed markers moving forward. And as I hope I highlighted today, is that data analysis is still a very active area of research using this data. So it's exciting, but it's also something that needs to be planned out and considered when moving into Codecs data. Thank you for your time. I'd like to thank Dr. Camilla Quirk, who's the computational biologist who did a lot of the clustering and the downstream analysis. Zara Yassin, who's a Codex scientist to run the machine. Oliver from Akoya did some of the initial staining when COVID first hit, and the single data was generated with a collaboration with Alex Warbrick from the Garland. Thank you.
Thanks very much, James. Wonderful work. And we're getting close to the end of the webinar time. So there's lots of questions coming through in the Q&A. If anyone has a direct question for James, either stick your hand up or stick it in the Q&A or the chat. What we will do is try to answer all of the questions that are in Q&A. I'm happy to hang around for a bit. I don't know if Danny and Giljay are happy to hang around and answer any questions as well.
Yeah, I'm happy to hang around for a bit.
Happy for that? Okay, so there's a few unanswered questions still in the Q&A. I don't know if there's any that are of interest there. I did take some notes of some of the questions that were coming in. If I can get my screen to work. So I've grouped them into 4 groups. I think we've answered most of the basics around why 40 markers, what the limitation is there. There's a few questions around staining. So what's a good general one? How well does the system deal with tissue autofluorescence, e.g. collagen or connective tissue? Anyone want to have a go at that?
I think I've answered that. So basically on the first cycle of the stain, it's pretty much DAPI with no other fluorophore. So that provides your background snap, and then you use that autofluorescence snap to subtract the background autofluorescence, if that makes sense.
Yeah, a lot of these questions have been answered already, Danny. I'm just going to go back through some of them just to make people kind of aware. Are you able to comment on how well Codex copes with rare antigens? They usually utilize amplification labeling system to visualize these antigens. So presumably that's rare and lowly expressed antigens.
We had some trouble with detecting particularly the checkpoints, which are even hard to detect with the TSA systems like the Opal system. So it is a problem with others, but it's a 1-to-1 without a major amplification in the Codex system. So PD1, LAG3, those type of markers we had struggled to detect. But it's not always the case, though. Some of the things that we normally see as highly expressed didn't work with the Codex system, but then some of the things that we struggled to detect with the Opal system worked well with the Codex. So I think you have to try. You don't know upfront which ones are going to work.
Yeah. One of the questions was, how long does it take to stain an entire section? I think
we've been doing it. It depends how big the image is the area is you image, but we were doing whole sections, and it's been for about 20 markers. It takes most of the day, but you don't have to be there at the machine doing anything, so it doesn't really matter if it's running over the night or how long it takes.
Yeah. In terms of analysis, so segmentation and registration is a little unclear. How are individual cells segmented and registered, and what's the precision of this?
We tried the Codex processor, which does cell segmentation itself, but we found it was missing a lot of cells, and we didn't feel that was going to be conducive to good spatial analysis downstream. So we converted to TIFF to put into the Codex, into the Halo system, and we did normal cell segmentation with a watershed method in Halo, and that was achieving good results. So it's no reason that the Codex data is any different to any other image-based data.
Yeah. And then finally, do we require to manually set a threshold for each marker? So the example given was CD34 in one of the talks. We know this may be different for each antibody.
And it also depends on what you're trying to. Some people, you could manually gate using the MAV software. We did try that, it works quite well. But we've been trying to do the approach where we go from the single cell to the Codex data. So using an unsupervised phenotyping approach, that's also possible, as Cam's work shows.
Okay, I think we're a little bit over time. We've still got a bunch of questions coming through. Let's see if there's any. Are there plans to move from the cover slip system to a conventional slide size? That's one for someone from Akoya. I believe that.
Yes, the short answer is yes, but we can't give you a definite time yet, but that's a work in progress.
What else? So there's a question again for you, James, around the amplification of PD1 and PDL1. Can you elaborate more? Where did you put it in the cycle?
So it has to be the last staining cycle because you can't strip off so it was a tyramide-based amplification. and it couldn't be stripped off, so it's the last staining run. And it uses, I think Akoya could comment, but I think they're going to be releasing these as commercial barcodes with the tyramide amplification system, but it's just barcodes with the tyramide system.
Okay. Does Codex work with old FFPE tissues, 5 to 10 years old? I guess your limitation there is probably whether or not they're going to be on a cover slip or not. But certainly from the blocks.
Ours were pretty old, so some of ours, because we're going through the duration of patient's treatment cycle, some of them were 5, 6 years old. But a lot of it, with the staining, a lot of it depends how long the sections have been cut for. So you really want to cut fresh sections the day you do the experiment. But we were using 5, 6 year old biopsies and they were working.
Do you see any change in morphology through the cycles as the cycles go on?
It's really good morphology compared to the heat stripping that we see with the opal system. So we actually see better morphology throughout all the staining runs compared to what we're used to seeing with our opal heat stripping technique. So I think it's quite good in that respect.
I think that probably covers most of the things. We'll continue to answer through the Q&A, and if I can work out how to download that, I will send that round to everyone after the webinar. Thank you to all the speakers, especially James for doing it live, and Giljay and Danny from Akoya for helping out answering those questions in the Q&A. As I mentioned, the webinar will be available again on our YouTube page, and I'll send a link out to that over the next few days. Might be the start of next week by the time I get around to that. But yeah, for those within Peter Mac, if you're interested in coming along and playing with the system, then please get in contact with me. For those within Australia, the part of the deal of the long demo is that we're a demo site, so you guys can also get access onto the system. I guess probably Danny is the best contact for you in that case. And for those across Asia Pacific, please reach out to your local Akoya representative. And with that, thank you, and we'll bring the webinar to a close.
Thank you, Paul. Thanks, everyone.
Thank you, everyone.
Thank you. See ya.